So my name is Iris Bika. I'm a Business Development and Licensing Associate here at the Center for Technology Licensing at Weill Cornell Medicine. We are the Technology Transfer Office for the Academic Medical College here at Cornell University. CTL is responsible for managing the intellectual property and commercialization partnerships for all of the technologies developed by university and medical faculty on our campuses, as well as our trainees. CTL is also part of a larger organization called Weill Cornell Medicine Enterprise Innovation, which provides education, programming, training, and mentorship to innovators who are interested in the commercialization journey. Today, we have the next installment of our special panel series, which is a series of webinars where we bring together panels of experts to address opportunities and challenges in the path towards commercialization. Today, we're going to be addressing the scientific, business, and regulatory considerations related to commercializing non-animal or alternative human model systems. So here at Cornell, we have researchers innovating new model systems to answer their research questions, as well as many more who will use these new suite of tools to advance their work. With the requirement for animal testing for new drug and biologic applications removed by Congress recently, we're edging closer to a different reality where these non-animal models comprise a much larger part of our scientific and drug development landscape. So today we'll explore the current scientific state of these model systems, potential paths and questions during commercialization, as well as the future of these systems in the regulatory process. So just some housekeeping, you can ask questions using the Q&A function at the bottom of the screen. We will try to address these at the end of the panel discussion today. Also, at the end of the panel, you will receive a questionnaire. We would appreciate if you could just take a few minutes to fill this out so that we can make sure that our content is relevant and meets the needs of our audience. And with that, I would like to welcome our panel and give them an opportunity to introduce themselves and also share their personal journey of how they got into the field of alternative model systems. So maybe start with Kristen while Lowry deals with this tech.
Yeah, sure. Can you hear me?
Yep.
Okay, excellent. So thank you so much for the invitation to be here today. I'm really excited to discuss this with all of you. I'm Kristen Bursack. I'm a principal scientist with a company called Mimetis. Mimetis is an MPS or microphysiological systems company. So we develop these advanced in vitro models of all sorts of different organ systems, vasculature, kidney, liver, brain, many of them. And yeah, the company it's a global company with our headquarters located in the Netherlands, while I'm based in our US office, which is in Gaithersburg, Maryland. And so there we're a much smaller team, about 10 folks, and I'm leading the R&D team of about 3 folks. And it's really our efforts to support the research and development of new advanced in vitro models that can serve the market, whether it be academic or pharmaceutical customers or pharmaceutical industry customers. And so maybe a little bit on how I got to this place. And I think it really started back in my postdoc work. And so my background is in toxicology. And before even doing my postdoc, I felt that I needed, as a toxicologist, to gain some experience in animal models. And so I worked on a mouse model during my postdoc, and I learned a lot. I gained so much experience. But that's where I really appreciated how important it is to help reduce and, the 3 R's, reduce, replace, and refine the animals that are currently being used in pharmaceutical research, especially. And so I was inspired to just be part of this movement that I think was really happening. And at the beginnings, maybe, 7 years ago when I was making that leap. And so that's when I just looked for opportunities to help contribute to that effort. And I found Mimetis, and I've been with them ever since. So yeah, that's a little bit about me.
Thank you, Kristen. Krista, would you like to introduce yourself?
Yes, can you hear me?
Mm-hmm.
Okay, I'm lucky, the lucky one. So I'm Krista Durney. I'm from Valo Health, and we're an AI-enabled drug discovery company. And so the way I came to Valo was by acquisition of a company named Tara Biosystems, where we focused exclusively on human cardiac organoids for drug discovery. Tara spun out of university, so it came from Columbia and excuse me, University of Toronto. I did my PhD at Columbia in tissue engineering, focusing on musculoskeletal systems, so not cardiac. And so just to, quickly translate into how I got to the space, I fell in love with tissue engineering during my PhD. I came from a mechanics background, and I thought it was just a cool idea to try and recreate organs in the lab. So that was, was purely from interest in biology and mechanics, that kind of brought me there. But then similar to Kristen, over the journey of doing research and experience with in vitro systems, in vivo systems, you kind of come to realize their shortcomings abound. And then there's technology just rising at the same time to enable us to have better human models that, can serve the purposes for research and, many different purposes that could improve our learnings from the, the in vitro systems and in vivo systems. So it seemed like a really good time during my PhD, over the course of my PhD, and then at the end to, translate that into a career. And I got really lucky because Tara came out of Columbia. So I was familiar with the technology, and I thought it was just incredibly interesting. And even though I wasn't in, the cardiac space, doing some research, recognizing that there was just a huge unmet need was a great place to start. And I also loved the opportunity to consider the business model. And so that's how I joined the team, was at the time of, okay, we have the science, we have the biology, we have the mechanics, like, what do we do with this as a company, as a business? And so, yeah, so that was just a great moment. I was super lucky to be part of that. And then, now at Valo, we're incorporating this into a whole other kind of rise of technology, where we're incorporating this with AI, machine learning to, come to a whole better cycle, or an improved cycle of how pharma considers identifying new targets, validating those targets, and then ultimately developing drugs. So it's been a really cool journey, and yeah, happy to be on this panel. Thank you for the invitation, and, looking forward to the conversation.
Thanks, Krista. Lowry, do you want to try again?
Let's try again. All right.
Yay.
Yes, nice. Technology worked the second time. Yeah, so Lowry Curley, I'm the CEO of Axosim. How I got into this was I myself had an health problem. So I had a seizure when I was in college. Everything's fine now, but along the way, I explored different novel ways of addressing the problem, and kind of came out on the other side of that. Number one, very interested in neuroscience. Number two, interested in translation, so getting the science to patients. And I had an opportunity to go to Tulane University to work on tissue engineering, focused on neuroscience, with a professor who had come out of Bob Langer's lab at MIT, so was very interested and focused on translation. And, a lot of things led to another. I founded Axosim to translate that initial platform and work on commercializing the product. So I can talk a lot more about that, but it has been incredibly fun and rewarding to have gone literally from the bench to now working with some of the biggest pharma companies in the world.
Great. Thank you all for those introductions and for joining. So since we have a variety of listeners joining, I'd just like to start with a brief overview. Let's see, can you see this? Examples of alternative, okay, great.
Yeah.
So I'd like to just start with a very brief overview of some of the types of technologies that we mean when we talk about alternative human model systems. And this is by no means exhaustive. So first, I think we're all familiar with 2D culture systems. This can mean primary cells, this can mean stem cells such as iPSCs, which have known drawbacks and are not always predictive of human outcomes. The next level of complexity is spheroids, which are simple 3D clusters of cells which can interact with each other and the surrounding environment, but don't ultimately closely resemble the structure or function of the organs they represent. Organoids, on the other hand, are more complex 3D aggregates of cells that can self-assemble, often resembling miniature versions of the organs that they represent. And these can include tissue-specific cell types, structures such as that which you can see here in this gut organoid, and functions. Another type is engineered tissues or 3D tissue models. which often involves configuring cells in a 3D structure to reflect the architecture and function of that tissue. These often leverage synthetic or biological scaffolds and also can use manufacturing methods such as 3D printing for generation of these complex patterns. Next, we have chip-based systems such as organ-on-a-chip. These are tiny devices that allow for more physiologically relevant environments for cell cultures, and this can include microfluidic designs to better mimic the dynamic conditions in vivo, such as blood or airflow, and they can represent an organ or potentially multiple organs. And finally, as an example, we have in silico models, which are computational models which can integrate artificial intelligence or machine learning and can be used to predict drug interactions and toxicities, find novel targets, and may even be able to represent entire people or clinical trials. So by no means exhaustive, but just some examples of the type of systems that we're talking about here. And now that we're on sort of the same page, so clearly there's a broad range of different modeling systems available to researchers, each with their own strengths, limitations, value propositions. Could you tell us a little bit about why academic or commercial researchers might choose one of these types of models over the other, specifically in the context of your own experiences?
Yeah, I can get started there. Broadly speaking, it depends on the organ of interest. That may sound obvious, but sometimes people come in having heard of 3D models and really don't know anything about it, right? So then they just ask, hey, how do I use this? And for us, we kind of ask, well, are you interested in neuroscience applications? And if they say yes, then it really comes down to what specific biology do you want? Because these cannot answer every question that you'd like them to, even if you are talking about the right organ. And there's kind of a concept that a lot of people talk about that fit for purpose. So it really is, what's your question? Then that's what biology do you want to probe? And again, for us, it's neuro, and it's evolved, which has been really exciting. Not easy, obviously, but exciting, where the platform that spun out of Tulane University, that was my PhD, we realized really it was focused on the peripheral nervous system and then motor neurons. We originally thought we could do everything with it, realized pretty quickly just the geometry, the methods, weren't as amenable to central nervous system. So then we licensed a platform from Johns Hopkins University that was more of an organoid model, and in this case had neurons, astrocytes, and oligodendrocytes. So this oligodendrocyte piece, I'll spare anyone who's not a neuroscientist, really it's critical to diseases like multiple sclerosis. So then we realized, right, that's our target market, right? Let's go after that. And now we acquired a company at the end of last year that brought us a new platform that's really focused on seizurogenic and epileptic disorders, which for myself was very interesting to get in. But in that case in particular, it was a very advanced model that we brought in that was ready to go. Because we've definitely learned, no matter the biology, getting these things to a point where they can scale takes a lot of work. It's one thing to build the biology, but I'll talk about in a little while the interplay between complexity and reproducibility as a key part of the whole conversation.
Maybe I can jump in here too. And I completely agree that it really, choosing the model totally depends on your scientific question. And for me, I think it's really important that you're solving a problem that you have, right? And that there's an advantage of using this model. I think it's easy to get caught up in the fact that these are these new and exciting models, and just throwing it in there, I think, is not going to be as useful as, do you have a problem that needs to be solved? And for me, what I've seen at Mimetis, I think there's a couple things that can be solved, but one example might be in our gut cultures. Typically, say you're a scientist and you have to understand, the transfer of a drug across a gut, and you need to know it soon. But typically you use a transwell model, and that takes you a month before the cells are polarized and before you can have this model to test the passage of your drugs across the barrier. While in the Mimetis platform, the organoplate, we can actually use perfusion, so flow, to stimulate the cells to basically polarize faster and be more differentiated, so that you would be able to run your experiment in a shorter time frame. And then I think what's also nice about microfluidics is it helps reduce the number of cells. So if you, the number of cells that you need to build the model. So if you have limited number of cells, limited amount of media, limited amount of compound, that could be another reason to maybe use a microphysiological system, organ on a chip system, instead of, you know, the standard 2D model that you were using before. But yeah, I think that's just an example, but it is really important that you're basically solving a problem and not just using it because it's cool, although it's cool to do that sometimes too.
I guess I'll jump in too and just say, you know, for cardiac, so just to reiterate, of course, organ specific. And so, you know, for cardiac, we know that human translation is really poor. And so specifically pharma, you know, struggles with this, where you go through a lot of activities in vitro, in vivo, animal systems. And then, you know, you come to a point where you realize, you know, you have these failures in humans that, you know, are catastrophic for like the investment you've put into date. So what, you know, you know, any of these systems, but specifically for cardiac, a human organoid or 3D system or whatever, anything that you could test sooner in a human or bump that up in the cascade, it's incredibly valuable, because it's giving you a sneak peek into like anything you might see downstream that you could maybe, you know, work on earlier and, you know, get it out sooner. So yeah, that's the one thing, is just that human translation piece is just incredibly invaluable. And I guess I'll just say from the Valo perspective, there's also a lot of value in connecting data and using human biology like this to validate concepts that come out of any of these, any types of modeling that we're doing. And so I guess I'll just talk about two things that we do. We do target ID using real world data. And so, you know, what that means is we're looking at, you know, healthcare systems and pulling out novel targets from actual, you know, patient data. And, you know, there's a lot that we do on that end that I'm not going to talk about today, but, you know, once you get to the point of you have a target that looks interesting and novel in a human, and you're like, hey, this might be beneficial to modulate in, you know, for cardiovascular disease. You know, what's the next step? And so one of the way that we insert our human organoids into this cascade is we do target validation activities specifically on those, you know, targets of interest. And so that just kind of, again, it gives you like a sneak peek into, is this real? And so I guess I'll just emphasize that, you know, it's always important to have biology at any, you know, and so right now we're relying on this, the whole field is relying on in vitro and in vivo systems to do that. And these human translational models just offer a more translational view onto how will this perform in an actual human. So it's saving us a lot on the back end, because we're able to test in a human sooner and get that peak. And I guess just one more thing that we do is our models are also chemical models. So we're using AI to model chemistry. So we're looking at all, you know, and that's just an enormous field. And so we have a lot of capabilities there. And then similarly, we have our model system. So say we, you know, come up with a novel chemistry, we can test it in our, you know, in our organoid and see, how does it do? Does it, you know, how does it interact? And it's different, you know, and the other cool thing about these systems is that they're isolated organs. And so you don't have to, you know, there's, you know, pros and cons to that. But in this case, you know, you can ask very specific questions about the human heart muscle and, you know, get that answer really quickly. And again, the cool thing about 3D organoids is that, you know, they're functional. And so the endpoints, we're going to talk about that later. The endpoints are really important. So it's what Lowry said earlier. You can ask specific questions about specific endpoints that are of interest to you. So, you know, that doesn't mean you can ask everything and everything of these, but it's really cool for specific things.
Yeah, great. Thanks for sharing that. And I think, I mean, it's really clear that there's a broad set of these systems, and they're very versatile, and they can answer a variety of specific research questions. And I think it's important to keep in the mind that while all of these are interesting research questions, not all of them are necessarily commercial opportunities. So how do you determine if your system and its unique features addresses a real unmet need? and what factors sort of influence customers' willingness to pay for these newer types of models?
I can, bring in sort of when AxoSim got started, a program that we went through that has served us kind of still, frankly. It was an NIH funded program called the I-Core program, or Innovation Core program. Essentially what it did was, force you, because it's a program, but get you to go out to customers and not sell your product to them, but basically ask them, what are your pain points, right? What are your needs? How are you filling them now? How do you need to be filling them? And then ultimately, would you pay for it? And as a scientist, it was really good to learn. Basically learn to listen in a sense. And just because you think it's cool scientifically doesn't mean pharma does. And some companies came out of that, or some technologies came out of that and realized it doesn't have a market, right? You have to have product market fit. So it wouldn't be a sustainable company. Thankfully, we came out of that realizing we do have a product, there is a market for it. And we learned a lot along the way, particularly we weren't selling the whole time, but we learned how to sell just by listening. And we've continued to use that. As I mentioned, we have 3 different products, multiple biological systems. And anytime we're exploring a new one, a new disease, whatever the case may be, we have ways of kind of asking customers, do you need, like, is it a big enough disease model, disease area to justify spending R&D on it? And then if so, like, what do you need, for it to be applicable? So that's something we continue to try to use, because there's just so many things any given one of these platforms can do. You really have to be focused. And that's really hard as a scientist, like really, really hard.
Maybe I can jump in next. And Lowry, we didn't do the I-Core program, but and I know it's intense, we are funded quite a bit by the NIH SBIR grants, which I think are a nice opportunity and really kind of nice transition, I think, from academia to these more commercially focused grants. And so through writing those, I also definitely learned a lot about how market research is important, and totally agree there. And also agree with you that not only is it enough, it's not only enough to identify this gap and this need, but do these folks have the funding, right, to be able to actually incorporate these new products into their daily work, and do they have the budget? So I think it's really important to also just really be talking closely with these groups and understand what the flexibility is with their budget, and if they have the flexibility to explore what is this really new space. And yeah, because I think, and we may talk about this a bit later, especially in the pharmaceutical industry, in the more preclinical drug development space, there's a little bit less flexibility with what models can be used, because a lot of this, most of this data, all of the data have to go into an IND for the FDA. And typically it's very regulated which assays and models are used. And so I think if there's certain budget for those models, then it may be that there's less budget for these more exploratory models. So more, perhaps a better or another space to think about them is in the drug discovery space, where there is more flexibility to look at different models. But I do think there's a place in the preclinical as well, and I think we'll chat a bit about that later.
Yeah, it seems like there's a variety of sort of different factors that influence the demand from the commercial side for these models. So I guess sort of speaking to this tension between the scientific process and commercial success, what are some of the scientific and commercial challenges during the process of translating these models that you had into a business context? I know Lowry and Krista, you both had sort of the experience of taking these systems from an academic environment.
Yeah, and kind of piggybacking off of what I said before, there's a little bit of, as a scientist, right, turned startup, turned business person, I guess you could say. I don't know if that's the right way of putting it. There's really a tendency to want to advance the tools, make them more complex, put in new biology, chase all these interesting things, and customers are going to ask for that. They want that. But what we realized is reproducibility is more important. So I'll kind of give you an example of kind of how we did this sort of through our first two platforms. The initial technology that licensed out of Tulane University based on my PhD again was really focused on the peripheral nervous system. So sensory neurons and Schwann cells. And we spent, 5 years of myself plus a couple of years of people smarter than me, as I like to say, refining that model with that biology. And so we spun it out. We had a decent amount of reproducibility. In hindsight, it wasn't that great. But then we tried to introduce, can we put motor neurons in this? Can we put astrocytes? Can we put oligodendrocytes in it? And realized like that we were just going in directions that reproducibility was not good. And frankly, we just weren't able to get clean data in the first place. So that's when we went and licensed the new platform from Johns Hopkins, which in that case got us into the central nervous system, multiple sclerosis type applications. Spent a really long time developing the methods to get that reproducible. And in academia, you certainly do this. So I'm going to be a little bit extreme in the way I talk about this, but you don't get grants, right? And your focus isn't, hey, can I do this thing 12 times in a row the same way? You want to be exploring novel things, novel biology. But we realize you have to do it the same way every time if pharma's going to pay for it. And so, then we got questions of, hey, can you put microglia on this? Can you make blood-brain barrier on this? Can you put all this new biology on it? And we could try it. And after, as I talked about before, first we validated, is there something commercial for it? And then if we started trying to do it, it was like, no, this is too complex. We have to focus on reproducibility. So this is just something we constantly ask ourselves and are very, very deliberate before we get into those things, because and I said it before, there's just so many things these platforms can do, but we have to take it one step at a time, or else you risk pharma seeing something and saying, oh man, this data is just not clean, it's not reproducible. We're not really into this whole organ on a chip thing in general. We really, because it's so emerging, I think we have to be really careful about how far we go.
Yeah, I guess I'll jump in too. And so I think, it's a great point about the complexity and the reproducibility, because I think that is everything here. And that's a key to, success. I guess I'll leave it at that. So I know for Tara from the start, so I wasn't there at the start. The beginning of Tara, the first 3 years were that. It was a full investment in, is this real? Can we reproduce this, what we've seen in academia? Is it something that we can scale and actually utilize? And, and I, I'm very impressed with that, strategy, because as Larry explained, like you can see how there can be a lot of twists and turns and pulls in a lot of directions. And I think that, the leadership then really did it right in, investing in that and kind of keeping it closed off. And, fortunately you have to have the funding to enable you to do that, to have the right team to, kind of just be heads down in, developing the platform and, the scalability of the platform. So, totally kudos to them for the years spent there. And, I was lucky to join at the point when we had something really real that was reproducible and that pharma, could utilize. And I guess I'll just say, during my PhD, I wrestled with the concept of, using tissue engineering as a therapeutic or, as something else. And I think that's common too, to consider. And I know for me anyway, my thinking was, it's so early and the FDA is so challenging, like, how do you get around that? And because I was focused on therapeutic, like I always thought that it just made sense, to implant these things in people. And obviously people are doing that. And, which organ is going to be the first, which, some are having success. And so, I was grappling with that. And, I was lucky to, when posed with the concept of going to Tara, the openness to, what if we're, a pharma decision-making tool? and I think that was a really smart idea, because there's just lower barrier to entry, and you can kind of work out all the kinks of what does this really do? What is this good for? And, and with the concept of a therapeutic always kind of on the side, but at least we can, develop the tissues, with a viable, way to continue to exist as a business. And so I think that was really smart. And so then, as Tara, we were effectively operating as a CRO. So there's also the concept of, products versus service. And, maybe we'll talk about that a little bit later. But, we were acting as a service because of the complexity of making these tissues and the time in the lab and how long it takes. We were, essentially doing this for pharma. We worked with over, 30 pharma academia partners, and we learned a ton about what do they care about, what could we do better, where should we spend time in R&D developing this. And so that was, an incredible experience. And I think, where we learned the two camps for cardiac anyway are, in pharma are safety and then drug discovery. And what we learned is that, drug discovery, they have the, or the largest kind of potential to explore these new organ systems. They're really curious about it. And in a sense, I guess I'll say, like, the FDA requires the other models, and, these models are a little bit, they're still on the cusp of being accepted by the FDA. So for safety, it's just a little trickier to, get for the groups there to get the funding to invest in these models. But drug discovery, on the other hand, they can see how they add value to their programs more quickly. And so, Valo recognized that, Valo was our customer at Tara. And so we were working closely with the folks at Valo when we were acquired, and, they loved what we did. They saw the value in the biology and how it plugged into, our AI, this whole ecosystem of, this highly translational human biology, and how it really enables drug discovery. And so, that's what led ultimately to us being acquired, and then now developing this partner focused business model, which is, essentially bespoke models for pharma. And so, we're for drug discovery. And so, we're not, doing these kind of, I guess I'll call them like one-off type transactional relationships. It's more bigger, deeper, longer term models that can be used for drug discovery programs.
Yeah, thanks. It seems like there's a lot of sort of decisions that have to be made in early days for finding what your final product is, finding the application. And I sort of want to expand on the last point that you were talking about, which is sort of finding your business model. So these are obviously very complex systems, and they sort of uniquely position you from a business standpoint. So speaking to that sort of service to product decision that you were talking about, what types of business models can be leveraged to commercialize these technologies, and how do you decide which one is sort of best suited for your model?
I guess I'll let someone else speak. Yeah, go ahead.
Yeah, maybe I'll jump in now, because I think at MedMetis we offer a little bit of everything. Like you were mentioning, Krista, at some point you kind of had to decide products or services. And at the moment we're doing both. And I think it's because we feel that we're meeting the needs of different types of customers. So I think we've found that, especially in the pharmaceutical industry, that's where we are having the more partnerships, like you mentioned, Krista, also just sort of smaller service projects. And it's especially because they don't necessarily have the lab space, and they prefer, right, to outsource the work and sort of work collaboratively together. But as you can imagine, the services tend to have a higher price point than the products. And I think a lot of the times academic groups, partially for that reason, partially because they want more control, would prefer to have the products in-house and working with them in their own labs to answer their interesting questions. And I think for this part, it is really crucial kind of bringing back the reproducibility part. It's so important that you're able to have a reproducible system, something that you can do as the expert in your company, but that anyone else can do in their lab as well. And I think I've seen a journey of MedMetis and a lot of learnings that we've had by interacting with the folks that are using the platform in their lab. And one thing that was really important and changed quite a bit was just the way that we trained folks. And, first it had to be an in-person training, and now we've kind of developed this more virtual webinar thing, and that also has time with our field application scientists. And so, yeah, we were able to kind of change based on the customer needs and to make sure that they would be successful in their labs. So yeah, we're a little bit of everything right now.
And I can tell Axosym's story in that regard. We, for most of our history, have been a service provider. And part of the reason for that, number one, when we spun out of academia, and there was still work to be done on the platform. We knew it was still a little bit early, but we had customers in industry who were ready to sort of kick the tires on it, let's say, with the understanding and expectation that it was still a little bit early. And part of when we were getting started, and you all should think about that too, because if it's a product and you ship this to us and it doesn't work the way it does in your hands, we may never come back. And that may not be any fault of ours, right? And so, that was two things. Number one, we waited, certainly to be a product. But number two, we could get paid to be a service early with the expectation to the companies of like, hey, here's where we are. If you're willing to pay to help us get closer to here, let's do it together. And so being able to get paid and learn from them, made it a no-brainer to just go ahead and get started there. But setting the expectations part is tough, because as a startup, as an entrepreneur, you're very optimistic, right? And you always think you can do more and more and more, but it takes time. And so that was another lesson learned, I would say, along the way there. But we do products now as well, at least with one of our platforms. And to get to that point, it took a lot of investment. And ultimately, once we thought we were there, we ended up basically doing boomerang shipping, where we would ship it somewhere, have them ship it back. Like, we did that a lot of times to make sure it was reproducible. And at that point, we had it where it was plug and play with some other commercially available hardware systems. So we worked with those providers to also put it in their hands a little bit, just kind of develop, I guess, Kristen, a little bit, well, like you said, kind of help develop some of the methodologies so that we were very confident it would work in our customers' hands, once we put it there. And then honestly, we kind of held their hands a little bit in the beginning to just work through every single step with them until they said, oh yeah, okay, now we're getting the same data. But it was setting expectations, the exact same thing I said before.
And Krista, what about you at Tower Biosystems and eventually at Valo Health? What are some of the business models that stood out to you at those places?
Yeah, so I guess I'll just say, we were always services because of the complexity of the system. It was not on our road. So maybe it was contemplated on our roadmap, but it's just, we decided that it made sense to remain in our hands to develop, and so I guess I'll just describe the system a little bit. So basically it takes us 7 weeks to mature the tissues, and that's a key to the success of the biology and the physiology, is that the tissues mature over time. And and because we're starting from iPSC, so we're starting with a nascent cell type, and it takes 7 weeks for them to to actually reflect the physiology of the human heart. And so that's kind of, and I know it's the same at Axosym and Mimetis for different things. Like this is the point, is that it's challenging. And so we stuck with services because I guess the big view, and especially because we were targeting large pharma and academia, is that that we constantly have tissues ready to go to test. And so we're saving you all of that, essentially 2 months of, monitoring these tissues, QCing them, making sure they're ready to go. And so we constantly have, the healthy tissues available for you to like run your screens. And so it's really, I guess attractive to pharma in that way when you present it like that we're saving you all of that, you don't have to have your perfect experiment designed and then you start and then you got to wait now essentially 4 months for a result, right? Like we're saving you a lot of time. So I guess that's also a key, is that there's such a tricky thing to navigate between how expensive these things are, how much time they save you. So like to come up with the real value, and then, how to make it attractive to pharma to kind of pay for it, when the value that they're seeing, it needs to connect to their own value drivers internally at pharma. And so that's why it's really tricky for safety. And I know we're going to get to the FDA part of this. Unfortunately, we're still not on the roadmap as something that is seen as a, an approved way to, validate or, to provide to the FDA as, something that says like, this drug looks good. I'll let the others kind of speak to it more formally, but yeah, so, until we're there, it's more difficult to get other groups invested in, we're still considered early, even though we're so kind of, we feel, very mature, very reliable. We have, everything kind of behind us to say like, look, this looks and feels way more like a human heart than anything that we have out there, including the in vivo models, especially in cardiac, the in vivo models, just, it's known that they're not great. We see failures constantly where it looks good, everything looks good. And then you go and you put it in a human and you see catastrophic issues. So, we're trying to, get there for, the safety groups. But so I think that's where that's why we landed where we are as a service provider, mostly targeting drug discovery. It was just a match between the value we create and then kind of where the budgets are within the pharmaceutical industry for where they're willing to explore more and, leverage these models to create insights and value for their own organization. So it's definitely, a tricky path to navigate to get all those things to line up, so that your company can still exist. You can continue to support, the business.
Yeah, and I guess another thing that I'm curious about, sort of in the midst of all of those decisions about products versus services, it seems like academic groups sort of share that exploratory mindset. Is there any room for collaboration with academia? Lowry, when you were departing from Tulane, was there any continuing collaboration with the group you were leaving?
Yeah, absolutely. Hands down, that is important, and we still do that because the exploration for where these things can go, the building complexity into them is so important, right? And at no point did I mean to, to speak badly about that, without a doubt, but with those collaborations, we're able to, gain insight and to provide some, platform, like literally provide platform, and then work with the academics, so work with Tulane, Johns Hopkins, the labs that originated these, and kind of be able to see and help with when we think they're ready for commercialization, right? So it's sort of these only exist, for the vast majority because they were developed, at universities, and the next iterations of them is the exact same thing, right? And then for the companies, for the translation, it's our responsibility, I would say, to see that and kind of say, hey, in conversations, we've now identified there's a market need. How is that going? Maybe we fed them some of that information to help them guide some developmental choices. And when it looks ready, engage their tech transfer offices, right? It's a very formal process to this. Go to their tech transfer offices and say, we're interested in licensing this. They probably filed a patent. So that's really important to be able to protect these. And then I keep using the word responsibility because it is, making sure pharma adopts these, what it works for one of our platforms helps all of them, right? And so then it's up to us to test and refine and develop the methods for the reproducibility part of that. So that has to continue 100%. And even new collaborations, right? It doesn't always have to be who you've worked with in the past or the labs that kind of originated these things, because beyond just the biology, you have to be able to scale it. And that requires a different skill set, right? Maybe that's more pure engineering or on the data analysis side. And so, we want to collaborate with the experts there without a doubt. It's just setting expectations with one another and kind of, realizing it's important to focus on your strengths and just have open conversations with each other.
I'll just say quickly to that point, where the currency of pharma is actually money, they have a lot of, that's what, they're huge in industry, academia, the currency is innovation and cool ideas. And so I think, when we were Tara, we constantly wrote grants and partnered with, academics who had really cool ideas to use our platform, to come up with something. And it was beyond just academia, but other, big groups, nonprofits that had, cool ways to work together. And we still do that because, we recognize that, yeah, like that's the tip of the spear with, the next thing and the coolest thing. And so we're, and we want to make our models the most utile, ultimately, for people to use everywhere. That's, the goal is that these need to, and I totally agree. And I know we'll talk about it later. It's just our whole industry is kind of linked arm in arm together on this, that like these models, any success for any of us is a success for the industry. And so, and any way you can, get at that is a win. So, partnering with academics, it's, that's just another way to showcase the possibilities and, how innovative, these platforms can be.
Yeah, thanks for that. And that's certainly good to hear. I'm sure many of our academics have models which they're either interested in translating or commercializing, or might be open to collaborations on existing ones. So good to touch on that point. And then finally, I want to switch gears a bit to talk more about the changing regulatory environment. And I'll just leave a quick reminder here. If you have any questions for the Q&A, please feel free to drop those in the chat and we'll respond sort of after this section. But for those that are not familiar, I'll first give a brief overview of the FDA Modernization Act 2.0, which was passed by Congress in 2022. So this act expands the range of model systems authorized for use in applications to the FDA to include cell-based assays and in silico models like those that we just discussed today. More specifically, it lifted the requirement for animal testing for all new drug or biologic applications. Importantly, it did not ban the use of animal models or change any of the current existing regulations at the FDA, which largely still requires animal data for successful submissions, but it did sort of open the door for this type of change. So I was wondering if you could tell us a bit more about what additional standards or frameworks need to be defined with the FDA in order to move towards inclusion of more of this data from alternative models into the regulatory process.
Maybe I can jump in first. And thank you for giving an overview of the Modernization Act. Yeah, I think when we saw this was passed, it was very exciting. There was a lot of buzz. There's going to be a place for us in the drug development pipeline and the regulatory side. But then what is the framework and how does it all work and how yeah. And I think one of the biggest questions now is who is going to define the framework? And is this going to be folks at FDA, at pharma? Is it going to be the people who are developing the technologies? And I think it's really come to light that it really needs to be everyone. Everyone that has a role here should be developing the framework. And so I think since the Modernization Act, there's been a number of consortia that have formed amongst those different sectors. The IQ MPS, the NA3RCs, are some that I can think of. And it's during those meetings that those folks start to define what the framework looks like and the benchmarks for including the alternative to animal model data into the IND. And I think one term that has really come to light through these discussions is something called a context of use. And so this is basically a specific application for your specific alternative animal model. So for example, I think it's not realistic for the FDA to say, this is what you need from an MPS liver model for an IND. So that's basically, it's not detailed enough. Instead, they might say, you know, these are the guidelines for an MPS liver model detecting drug-induced liver injury of small molecule drugs that are suspected to have drug-drug interactions. So like very, very specific, you kind of take your idea and bring it down